Let's get started. So Dr. Carey, let's start with you, and maybe just to level set, and helping make sure everybody really understands all the various terms. So first, how do you define precision medicine, and how do you apply precision medicine to your research and to your clinical work with patients?
Well, precision medicine is, the short form is it's the right drug for the right patient at the right time. So it's basically using tools, and increasingly these are genetic tools, these are molecular tools, to much more clearly define the biology, typically of the cancer and of the person who has the cancer, to figure out how to tailor therapy more effectively than just giving everybody the same regimen according to a cookbook, which is, when you don't have these tools, that's what you end up doing. So that's essentially what I think of precision medicine as. How do you use it? I think breast cancer actually was one of the first precision medicine entities in the cancer universe, because we started using the presence of the estrogen and progesterone receptors and the tumor as a guide for whether or not the tumor would respond to anti-estrogen therapies. And then subsequent to that, now, decades ago, we started using HER2. Actually, HER2 was first used as a tool to identify chemotherapy benefit. But then, of course, it became the tool that identified whether anti-HER2 therapies would be useful or not. So we were several decades ahead in the precision medicine initiatives. But of course, now things have changed and become much more sophisticated. And we're still learning how best to employ many of these tools, but we'll talk a lot about this, but that's in general how I would think about it.
That's really helpful. I think often we forget that tamoxifen was really the first precision medicine. And I know it's hard for those patients that are triple negative and don't have a marker. So thanks for that overview. So let's pull Dr. Wagle into the conversation. And maybe you could talk about how do you define precision medicine today, and how does it look different than it did, let's say, 20 years ago?
Great. Well, first, thanks for inviting me. It's a real pleasure and real honor to be part of this conference again. I think precision medicine is, I like to take, as Dr. Carey just said, I like to take a very broad view of what precision medicine is. And it really is identifying features of the tumor or the patient that helps guide our decisions about therapy. And I think it's important to start with that definition, because if when people are having conversations or debating about precision medicine, everyone is using a really different, but sometimes narrow definition of precision medicine. And that makes the conversation, that can make the conversation confusing. So precision medicine today, I think, builds on what we've learned over the last several decades about the estrogen receptor and the progesterone receptor and HER2. But now, with the advent of lots of new technologies that let us interrogate every individual's tumor and look at the specific mutations that are present in those tumors, it helps us tailor therapies even more. I'll just add that we're still in the early stages of understanding this, and there's a lot more work to be done. But the promise of precision medicine is that we will be able to identify even more features of tumors beyond the big categories of subtype.
That's a really good overview. So Dr. Carey, maybe you could jump in and talk about how this has changed your approach to treatment.
No, I think, the truth is that, we were using this already, years ago to drive treatments according to HER2 and ER. Then in early breast cancer, I would say the next big advances in breast cancer were in the early setting for ER positive, HER2 negative breast cancer, with the development of all those genomic tests that help us determine who might need or might not need chemotherapy added to anti-estrogen. So that was fairly transformative for that fairly somewhat limited group of patients. It's not extrapolatable to the metastatic setting, but it is the kind of thing that I anticipate we will be having more of for the metastatic setting, and more of that kind of analyses in the future. I think for metastatic cancer, if you think about it, we've been talking about the genetics of the tumor and tumor-based tests. But the truth is, one of the biggest changes in terms of treatment for metastatic breast cancer, particularly as you mentioned earlier, triple negative, was the idea of using immunotherapy added to chemotherapy. And immunotherapy, of course, is part of the host. And what we now look for is evidence of immune activation around the cancer as evidence of whether or not immunotherapy is going to work. So that's actually been a big change, and one that's, to be honest, it's changing every day now in terms of what tests we can use, optimizing those tests, which drugs are going to be appropriate. A little bit of the Wild West at the moment. We have several tests and there's several drugs, and they're sort of how this is going to play out, I think, still remains to be seen, but there are several good ones. And so I would say I categorize the way these things are used now, and with a focus again on the metastatic setting. We do tumor-based testing, and Nick talked about mutational testing. We do that for the use of PI3 kinase inhibitors. We also do testing for evidence of immune activation for the addition of immunotherapy in triple negative. And that's the kind of thing. And I think that duality of looking at the host and looking at the tumor is going to be the way that we get better at this. We have a long way to go, though. We're not close yet. But I do think there are a number of really good initiatives going on that are going to help not just certain subtypes, but all of them.
Thank you. So Dr. Wagle, how, something we want to always know is, is this available to everybody across the country? And if not, what are the barriers, and what are we doing to try to change that?
I think that it's a really important question, and the question is what's available, what are the barriers, and how do people use them? And I think if you think about Dr. Carey's distinction there about some of the things that are more established that we better understand how to use, and compare that to the things that are newer. They're different. The availability of those two things are different. So let's start with the testing that is part of now standard care or more routine care. Those involve things like the receptor testing and looking for HER2, and now looking for PI3 kinase mutations. Those are tests that should be able to be done regardless of where you get your care. Now, when you talk about the kinds of sequencing that people often think about when they're talking about precision medicine, which is getting a test on your tumor that looks for mutational changes across many genes, many hundreds of genes, the kinds of things that are commercially available or available at many cancer centers, that is not something that's necessarily widely available. It's not something that's necessarily covered by insurance. And part of the reason for that is that in many cases, that's still experimental. And so when we think about those kinds of tests, for the most part, we think about them as helping us understand experimental therapies or clinical trials that we might want to try. There are a few exceptions where those kinds of tests can also tell us important information for FDA-approved therapies, and Dr. Carey mentioned a couple of them: PI3 kinase mutations, immunotherapy, and rare fusions that are present in many cancers, not just breast cancer. But for the most part, the information that we learn from precision medicine tests like tumor mutation profiling are ones that help us think about clinical trials and experimental therapy.
That's great. Thank you. Janine, do you have a question from the panelists?
Sure. We have a couple of great questions. The first one is whether you think that all people with metastatic breast cancer should have genomic testing. You kind of started to talk about this, Dr. Wagle. Yeah,
I'll share my view, and maybe Dr. Carey will as well. I think that there is a clear role for testing of the tumor when we're thinking about clinical trials. At the same time, we have a number of standard therapies that we use initially in metastatic breast cancer that don't require genetic testing. And so the question is really where in the spectrum of metastatic breast cancer, and what treatments have already been used, and what is being considered, our clinical trials being considered to determine whether or not genetic testing is going to be important. And I would say, I personally believe that there is information, important information that can be gleaned from a genetic test like that, but it's important to know what you're going to do with that information. If there has to be some availability of clinical trials or a physician who can interpret those results and then point to a right therapy. I think right off the bat, we don't necessarily have treatments that we would use based on the results of a genetic test of the tumor initially in metastatic breast cancer.
Janine, I think, Nick really highlighted it very well. I'm going to separate out a couple of things that I think are really important. When we talk about genetic testing, there's two kinds of genetic testing. Yeah. There's genetic testing of the person, right, which is the genes you inherited from mom and dad. And I do think that there is particularly in breast cancer, there is a compelling reason to do genetic testing in metastatic patients. And that's because we actually have drugs that work specifically in patients who have inherited kinds of breast cancer. And what we've learned over the years is that more people who didn't obviously fit into the family history categories and other triggers that might have caused us to do that kind of genetic testing actually might carry one of these mutations. And I think there's a reason to think about that. Then there is testing of the tumor for what's typically mutations and an abnormal number of genes. So it's really DNA, if you think about it, the blueprint stuff that they're testing. And I, like Nick, put those into two categories: things that we know what to do with, and things that we think that we know what to do with, but we're not totally sure. And actually, there's a third category of we have no idea what to do with these mutations. We don't have a drug for them. We're not quite sure what they do. There's a whole bunch of genes that have these weird names that mean that nobody knows what they do. And I think so. The first category is kind of an easy one, right? The second category is the one that troubles me more, because when you do these broad tests, you come up with things that are, for example, mutations that happen in melanoma, and that we know if you give a certain drug to a melanoma patient with that mutation, the drug tends to work. And what we have learned, unfortunately, the hard way, is that if you find the same mutation in a colon cancer, the same drug may not work the same. And the only reason you would know that is if you actually collected information about how patients behave. And the way that happens is through participating in trials. And there are a lot of trials that are looking at trying to collect the data about, how did patients do when you tried these things? And I think the FDA has been remarkably nimble over the past few years, much more so than I think they were, many years ago, about allowing things to move forward and allowing people to receive drugs fairly quickly once there's been proof of effectiveness. But I think when there isn't proof, it's really important that we make sure we're collecting information so we don't have people going down rabbit holes of treatments that don't work and aren't ever going to be proven whether they do or not because they're not part of a trial.
Can I add one more nuance to that? So I agree that in the realm of findings that are not already FDA approved, a lot of them may be involved with things that we are extrapolating from other cancers, and melanoma was an example, right? We might find a gene mutated in breast cancer that we know works well, that is present in melanoma, and the drugs work well in melanoma, but we have no idea if they work in breast cancer. And there was for a long time this idea in precision medicine that the tumor type doesn't matter, only the mutation matters. And if you find the mutation, you can use the same drug regardless of what type of tumor that the person has. And that's turned out not to be true. And so it is much more nuanced than that, where both the underlying genetics and molecular features of the tumor as well as the tumor type matter. And so a mutation that you see in melanoma versus colon cancer versus breast cancer, those three, even if it's the same mutation, those three tumors will behave differently. So it's really important to know that. But I would say that there's another category of findings that don't lead to drugs that are currently FDA approved in breast cancer, but they really are breast cancer specific findings. So there are a number of things that we can identify in breast cancer that we think are going to lead to, as Dr. Carey said, that we think we know what to do with it. not because they're present in some other cancer, but because they're important in breast cancer. A classic example is ESR1 mutations in the estrogen receptor, or mutations in HER2, or a gene called AKT1. And these are findings that are kind of on the cutting edge, and clinical trials are being tested in these subtypes.
I think we lost Dr. Wagle. So Dr. Carey, I'm actually going to follow up with another question from one of our participants that we're sort of starting to get to. So a number of people are asking about, there's a lot of different kinds of these types of genomic tests, or another word that we've used and that LBBC has been involved with a collaboration to kind of talk about the vocabulary, where we've been calling it biomarker tests. And I'm wondering if you could talk a little bit about how a patient can know which test to ask for and differences among the patient, among the tests.
Well, typically, it is hitting a moving target a little bit, because the biomarker tests are themselves evolving. And let me just start by saying there's some general principles about what we call biomarkers. And biomarkers are everything from ER to these super complicated, every gene in the universe type tests that Nick was talking about. But all of them have to have the same fundamental principles applied to them. Number one, you have to know that the test itself has been what's called analytically validated, which means that when you do that test, it actually behaves the same every single time, right? If somebody creates a test in their garage and, one day when it's 89 degrees, it behaves differently than the next day when it's 42 degrees, that's not an analytically valid test. So there has to be that process. And these are the kind of things that the FDA insists upon. Then you have to decide whether it's been clinically validated. And that's the crucial step when it comes to biomarkers as it applies to, for example, breast cancer. Meaning, does it separate the cancers into meaningful populations? So use the ER again, ER positive and ER negative. Is that a meaningful distinction? And of course, we know that it is. The last one is clinical utility. And clinical utility is, if you know this information, if you know the result of the biomarker, does it actually help you treat the patient better? Does it help the patient have a better outcome because of the use of that biomarker? So that's the final step. I mean, there are many tests that will segregate tumors into different types, but if there isn't anything you can do with the information, then it's not clinically useful. So that's those three steps, which is what's required of all of these tests for payers to pay for them, for the FDA to approve them, are actually super important. And so that's why typically when, most oncologists actually know, for a particular setting, for a different clinical subset of breast cancer patients, for a certain setting of line of therapy, et cetera, typically they know what the clinically useful, the ones that have passed that last test of clinical utility, what they are, because it has to do with drug choices and with prognosis or prediction of therapy. Does that make sense?
It
does. Did that answer the question?
Yeah, I think it does. I know everyone wants to hear the end of Dr. Wagle's thoughts. We'll bring this back to him, but then if Dr. Wagle, if you have internet, and if not, Dr. Carey, you can answer this. If you can also talk about circulating tumor tests and sort of bring that into the conversation as well.
Yeah, well, we're going to try this without video to save on bandwidth. So what I was just saying is that in breast cancer, there are a number of things that we know we can find on with tumor testing that don't lead to FDA-approved drugs, but do lead to pretty well-established and mature drugs that are not yet approved and are being tested in clinical trials. And I would say that's another category. And the examples I have given are ESR1 or HER2 mutations or AKT1 mutations, where there are drugs that target those mutations. PI3 kinase mutations were in that category as well until very recently, because the clinical trials were completed and showed that gene mutation was successfully targeted by drugs that target it. And now that's an approved therapy. And so we may see over the coming couple of years several of these other genes that are frequently mutated in breast cancer essentially graduate from this category to the category that of FDA approval. And so I would say when clinical trials like this are available, this is another option. It's not necessarily that we have to make extrapolations from other cancers. There is a lot of good clinical science and preclinical science that are working on these genetic alterations in tumors that have led to really important hypotheses about how we use these new drugs. And the second question was on circulating tumor DNA. So this is a really exciting and really powerful technology that allows us to obtain some of this information around what genes are mutated in a tumor without having to do a biopsy. And it's by looking at a blood sample and being able to see small amounts of DNA that are being shed by the tumor into the bloodstream and are now able to be sampled with a simple blood test. The advantages of this are it means you don't necessarily need a fresh biopsy to be able to get a snapshot of the tumor in the moment. And since we know that tumors evolve over time, looking at the most recent picture of the tumor might be useful because it tells you what the tumor looks like right now. There are a number of things that cell-free DNA or circulating tumor DNA can't tell you. And particularly, we can't learn about things like the estrogen receptor or the HER2 receptor as reliably by looking at cell-free DNA. So there's still, getting a biopsy is still important for some of the very routine things that we want to know about a tumor, but there may be additional information that we can get from cell-free DNA. The only thing I would add is that like we've said already for many of the other things, cell-free DNA is still in the early days. And there's a lot that we don't yet understand about cell-free DNA. And particularly, there is an issue where we don't quite understand both false positives and false negatives. So sometimes you find something in cell-free DNA and it may not be biologically or clinically meaningful. And sometimes you get a cell-free DNA test and it misses things. And so we're still really learning about how to interpret cell-free DNA. And there are a lot of clinical trials that are starting to look at this. And I think we're going to get a lot more information in the coming couple of years. I believe that cell-free DNA is extremely promising, but we do still have a fair amount to learn about how to use it.
Thank you for that, Dr. Wagle. And sort of as a follow-up to that, whether your results are coming from the cell-free DNA test or from NGS testing, if you find that there's a mutation that doesn't have a clinical utility, but that matches in another cancer type, that say there's an approved drug in another cancer type, one of the things that we hear is that people aren't really sure, should they try something that hasn't been tried in breast cancer because there's a match, or should they stick with a standard treatment that's approved for the disease? And I'm wondering, Dr. Carey, if you want to speak to that.
And can you also define cell-free DNA? Because there's a lot of questions.
Oh, sure. So cell-free DNA actually is what it sounds like. So instead of, there was a technology that was developed a number of years ago called circulating tumor cells. And you would find cells that were actually whole tumor cells floating around in the body. And those were used, basically, they tend to track with the amount of cancer there is in the body. They didn't prove to be as useful in terms of directing therapy as I think originally hoped. But cell-free DNA is just the DNA of the tumor, which you can find in microscopic amounts, and reflects the, one hopes, the genetic makeup of the tumor at that point in time. And I think what Nick was commenting on is super important. I mean, one thing it can allow you to do, particularly once the technology is better developed, and it is, to be honest, it is a bit in its infancy, is you can do what's called a liquid biopsy by just drawing blood, as opposed to needing to biopsy the tumor. And right now, you do still need to biopsy the tumor for certain things, but we are evolving towards being able to get much more relevant information out of the bloodstream than requiring people to have to go through biopsies, which we all would prefer not to. The second is, and this is I think what he also alluded to is you can see changes in the cancer over time. And we know that cancers evolve, right? Cancer evolves from the primary to the met, and cancers after becoming metastatic can also evolve. And sometimes that's in clinically meaningful ways. And the example that he used, which I think is an excellent one, is the development of ESR1 mutations. But there are many other things that can change. And in some tumor types, leukemias being more developed in this realm, they actually can see the impact of the drugs that have been given in driving the tumor in a certain direction. Whether or not you can tailor therapy to that over time, I think is a possibility, and one that I think people are hopeful. There's a lot of studies going on about that. But I also think that Nick used exactly the right word when he said, these are tests that may help with treatment decisions. And I think right now we are still in an evolution about how these can be used and how best to do them. But they're hugely promising for the reasons I just said.
Thanks, Dr. Carey. And I used a term when I asked the question. I said NGS testing. Would you mind redefining that, please, for our audience?
NGS stands for next generation sequencing. It's actually not at all important that you remember that, and we'll stop calling it that. It is a short form, and it just means broad mutational testing for typically, and originally when it started, it was abnormalities in the DNA, either too many or too few of the genes that you expect to be there, or mutations in them so they become different.
Thank you. And I think it's a good example of how confusing this can be, because we use so many different kinds of terms to describe these kinds of testing. One of the questions that we're getting in lots of different ways are from people who have had genomic testing at some point, maybe a few years ago, and are asking, should I have that testing repeated at progression to look for more different mutations? And can the old tests be useful after you had a progression?
I think, well, it depends on the nature of the test. So there are certain tests that have been around for a number of years. They haven't changed very much. I think, people can have some reassurance that they had a decent test done. If you had let's say, for example, if you just had, in the olden days, we talked about the test for inherited breast cancers. Many, many years ago, the only thing they knew to test for was BRCA1 and 2. Now they have a much broader panel of things, and some of them have proven to be particularly important. And while they're rarer, having a repeat test of those kind has actually already been shown to be useful and is quite routine now. I think from the next generation sequencing, NGS, going back to that, so the more broad tumor-based testing, many of them had a pretty good handle on the genes that might be relevant, from when they first started, because they're actually fairly recent. I think the question of whether redoing it is a question we really don't know the answer to yet. We do know that cancers can evolve. We keep using that example of ESR1 mutations, which are clearly mutations that are driven by the cancer adapting to being exposed to anti-estrogens. So it is an acquired mutation. It is rarely found in cancers that haven't been treated with anti-estrogens before, but then it does evolve, and in metastatic disease, it can be about 30 to 40%. And it can alter effectiveness of anti-estrogens, but we don't really know how to leverage that yet. That's an area of great interest. So I think it's good to know, but it doesn't hugely change the choices of therapy at this point in time.
I'll just add, I think that, the question of whether to repeat a test could either be about whether the test is different or better, or whether the tumor has changed. And so I'm not sure which of those two aspects the questioner was asking. But I think in part it depends on what is where in therapy and what are the options being considered. If there are, if you've had testing of your original tumor, but now you have a metastatic biopsy that has never been tested, ESR1 is one example, but we know that breast cancers evolve quite a bit, and there are these changes over time. But again, we're back in the position of even when you see changes over time, the question is what to do with them. And so for clinical trials, for the use of therapies that might overcome resistance, looking at the newest possible biopsy, whether that's a biopsy or a blood sample to look at cell-free DNA, I think is really helpful. But it's only as helpful as what your options are, and the options often are the availability of clinical trials. And I would say this brings us back to an issue we haven't really brought up and haven't talked about, but it's the availability of clinical trials and the eligibility of clinical trials, and making sure that these kinds of experimental therapies are available to more people who are getting their care at more institutions. And it's really that problem that has to be addressed before we can address the full value of being able to test these tumors.
That's a really good point. Thank you. There's so many questions as always, and there's a lot of really specific questions. So Janine and I are going to do our best to generalize them, but I'm wondering, Dr. Carey, given how many tests there are and how this seems to continue to change, do you kind of have like a playbook, sort of like these are the most important tests to get, and how can patients just start to move through that? But also we're getting questions from women who are in different parts of the country and maybe have community oncologists that aren't as expert and don't really have the time to do this, sort of help them with this process. And so what do you recommend for those patients?
Well, I think the playbook, again, is not that complicated at this point. I think the playbook is pretty well recognized for breast cancer and for other cancers in terms of things that we know affect clinical care right now. I think and as we move towards clinical pathways and things where we have all these guidelines that are out there, and Nick contributes to them, I do, everybody does, where these are nationally and increasingly internationally. So we have different guidelines for low and middle income countries than we do for high income countries about what kind of tests do you need to have? How do you interpret these results? So those are out there, and those are available to community oncologists, blessedly. One of the nice things about both electronic health records and the proliferation of communications tools that are online, they have access to these, to what test to order and what to do with them. And to be honest, when they don't know, they usually reach out and contact, I get contacted by people all over the world about what to do about certain things. And I think that's frequently the way. But I am not that troubled, and particularly in breast cancer, that people don't know what to do with information that comes in right now as it pertains to clinical choices that we have in the tools in the toolbox at the moment. I think the harder ones are that middle group that we were talking about earlier, where they get a result, it's an unexpected result in a breast cancer patient. They're not sure whether there's been, tests done on this particular mutation that maybe you usually see in melanoma, and now you find it in the breast cancer patient. Is it okay? And that's where I think the trials have to be part of this. That's where I think people need to have access to that tool. And the community oncologists, a lot of times they can also refer people. And I would guess, this is a maybe pie in the sky, but I would guess that some of these things will become, through the use of real world evidence and data that's collected much more broadly, we'll be able to kind of gather information from people being treated, even if they're out in places where they have no access to an academic setting. But I can tell you, the oncologists do have access to academic doctors. They have access to people who are expert in this. And so I always tell patients, I'm not so worried that they need to be treated, physically at a major quaternary center. I think that can be helpful, and particularly to touch base intermittently. But, our community oncologists typically are very, very good, and they know how to pull the trigger on finding out more information. As this gets more complicated, it's going to get harder for them. And so we are going to need to build better networks and build better communications tools, because I think we're doing okay right now, but I can see the future being much harder for them. And I do think that we need to put some real pressure and focus on that.
And I would say,
thank you. I just want to say that's so helpful. And I hope everyone is hearing that your community oncologist does have access. And I know because of the coronavirus, we don't know how long travel may be really difficult for particularly the metastatic population. And so just to tag on to that, like, are you doing second opinions via telemed? Is that something that is possible now? Sorry, Dr. Wagle, I didn't mean to cut you off.
No, you said what I was going to.
Okay.
The short answer is yes. I'm doing all of our second opinions are done virtually now. And I think that's to for a lot of really good reasons. Bringing patients fewer touches with the healthcare system is better for them. And so I think I've done one in-person second opinion in the last 3 months.
Okay. And the insurance is covering that?
Insurance is absolutely covering it. They've been very good about that.
That's great. Janine, do you want to grab another question?
Sure. Another question we're getting is that it's very common in metastatic breast cancer to have disease in more than one organ. And should you try and get a biopsy in more than one organ and have that cancer analyzed in both organs, and what to do if there's differences, genomic differences between the two biopsies?
Yeah, I'll let Nick answer that, but I want to say one thing that's really important that I fear I may have misled with my last answer. I have done a ton of second opinions. I can't do a second opinion for a person in Canada, which was a request that came in, because I am not licensed to provide care, so we are restricted to within our, like I have to give second opinions to patients in North Carolina who have, so we do have some things that you have to be attentive to that are rules, but I'm saying within what I'm permitted to do, that's certainly possible. My apologies if I wasn't nuanced enough in the answer about that.
So this is a really hard question, a really important one. And the answer is we don't typically biopsy more than one site because every biopsy carries with it some risk, some discomfort. It's hard to get a biopsy of more than one site at a time. And second, because we wouldn't really know how to interpret that information. It's true that cancer that is in different sites of the body at the same time may look different, right? If you did a test, whether it's an ER test or a HER2 test or a genetic mutation test on two different biopsies taken at the same time from different parts of the body, they may look different. And there are plenty of examples where there is what's called tumor heterogeneity, where different mets in the same patient at the same time have diverged and look different. But clinically, we wouldn't know how to deal with, whether we should follow the thing that we see in the liver met or whether we should follow the thing that we see in the bone met. And so it is a limitation of our current way we think about cancer. But what we typically do is when we do a biopsy, we make the broad assumption that most of what we see in a biopsy is present throughout the body. We make a treatment decision, and then we see. And if it turns out that one tumor somewhere in the body didn't respond as well to the other tumors in the body, then we have to think about what to do next. The one sort of way that we're starting to overcome this is this concept of the liquid biopsy or the cell-free DNA that we were talking about earlier, because the expectation is that because this DNA is circulating in the blood, it's a combination of the DNA from all of the different tumor sites in the body that are kind of being collected in the blood. And when you profile that, you are getting a snapshot of what all of the tumors in the body look like, not just where you biopsied. And that's one of the real promises of this technology. And one of the reasons people are excited about it. But that idea, although we have some good evidence that that's true, is still being confirmed.
Let me just add something. I think Nick outlined it beautifully. I think there's some reassurance from data that have come from, patients where they did test multiple different tumors from different places. And while we don't, we can't be too definitive about this because these are small series, it does look like the clinically actionable important mutations, the things we call drivers, right, and the things for which we typically have treatments, those tend to be shared broadly. They tend not to show up in just one organ. So I think there's some reassurance in those kind of data.
I'm wondering, either one of you could answer this question, but a couple of people, and we're getting this a lot at Living Beyond Breast Cancer, which is, what is the impact of COVID-19 on the progress of clinical trials and finding new treatments? So I'm happy for both of you to address that.
Yeah, so, I'll start. Yeah. Early on, and when we didn't know how bad it was going to get, and there was a very real risk of running out of beds, running out of ventilators, like really hard decisions that might be faced. And God only knows, we could still end up there, but it looks like we're not going to get there, blessedly, or at least we have the tools to avoid that. But early on, we put a lot of restrictions on clinical trials. And we also put a lot of changes in how we handled patients interacting with the healthcare system. We minimized how often they came in, right? Which of course has implications for clinical trials. If there are trial related things that have to happen coming into the healthcare arena, if you had to have a research biopsy, we weren't going to bring a person in and put them at the risk of being affected by COVID-19. And we needed to spread the patients out. We couldn't have packed lobbies full of patients, right? We had to create social distancing. We had to create new systems for making sure that they were screened and they could proceed safely throughout their visit and minimize interaction with visitors and with people who didn't have as high a need to be there. So there was a lot of emphasis, which really shut down to some degree clinical research, not entirely. It went down briefly by about 80%. We always allowed patients to enroll on trials where the trial was clearly in the patient's best interest to participate in it. If in fact this gave an opportunity for something that they couldn't get otherwise, even if it required a bunch of extra visits, that was permissible. Or if the trial was designed in such a way that they could be seen and treated on the trial using the same number of interactions that they would have had to have had they been treated with standard of care. So those always went on. I can say that, and all the strategies that we built to protect the patients and to help them navigate through the healthcare system safely are still in place. They're going to remain in place. We are now opening up, and have been for the last several weeks, some of the systems for allowing the trials to get back up and running to the same degree that they were. And we're seeing some promising evidence that they are doing that. And we haven't seen a bump in, we haven't seen a single instance in my institution of COVID-19 that was transmitted in the setting of the healthcare operation. So I think there's cautious optimism that we did put some safeguards in place. We may have overshot the market. I think that's okay for a month or two. And now we are kind of judiciously opening back up, at least at my institution. And I think also at Dana-Farber, but Nick can speak to that himself.
Yeah, I agree with that. I think there's no question that the last several months have been really hard for patients with cancer, particularly those with metastatic breast cancer and metastatic cancer who often rely on clinical trials for their care. And like Dr. Carey at Dana-Farber, we had a hold on most clinical research for the last several months. And we're in the Northeast, as things have improved tremendously over the last couple of weeks, we're starting to reopen and starting to increase the enrollment on clinical trials at our institution as well. I would say that I expect that things won't be as bad as they have been the last 3 months for patients with cancer, because as Dr. Carey just said, we are learning how to deal with this. But also there are going to be continued hardships and continued impact of COVID-19, both in the clinical research realm as well as in the basic research realm, right? There are a lot of issues where labs shut down and funding has been limited or cut. And I think I really liked Jean's comment at the beginning about metastatic breast cancer doesn't quarantine, because I think it's really important for all of us in the field who do research on breast cancer and who care for patients with metastatic breast cancer to realize that we can't we can't just pause, right? We can't just stop. And I think all of us in the community have been thinking hard about how can we adapt to this new COVID-19 world to make sure we don't slow down what we are doing both clinically and on the research side for patients. And I think we're starting to emerge with a number of new approaches and new ideas to make sure that continues.
That is reassuring to hear it from both of you, because I know for everyone listening, this is a really big issue. And I do think we've changed as individuals about our willingness to travel and take other, we all have our own sense of risks. So I know we have to keep on time. That is our mantra at LBBC. I want to give both doctors a chance to just say a few words about, or what you see as the future. And then Dr. Wagle, if you want to say anything about the Metastatic Breast Cancer Project, even if it's just to let people know where they could learn more, I think that would be really helpful. So Dr. Carey, why don't you go first?
Oh, sure. Listen, I think that, COVID-19 notwithstanding, and I think we will be back up and running, and we will have ways of doing this safely to whatever degree we can. We can't eliminate it, but I think we can create processes that work. We will be back to trying to make things better. I've been around long enough that I can attest to an amazing trajectory of therapeutic changes for metastatic diseases of all kinds, but in particular breast cancer, with absolutely tangible changes in outcome. We're not where we want to be, but I'm encouraged by the fact that no one's letting up, right? People's feet are still on the gas, and the recognition of the importance of this remains. And there's all sorts of really relevant things that we're only now, again, we talk about the tools in the toolbox, and we've spent the last hour talking about largely testing DNA, the blueprint of the cell, for abnormalities that might help us treat people. What we haven't talked about is all the newer tools to look at RNA, which is, the next step from DNA, and proteins, the things that are actually made and are functioning in the cell. How do we test for those? And there's a whole bunch of people who are doing fantastic work. It's not yet commercial or clinically available, but it is, there's the old line about the person, the drunk looking for his keys under the lamppost, and the policeman comes, he says, well, is this where you lost them? He says, no, but this is where the light is, right? So, people use technology, and if you have limited technology, you're going to keep using that. But the technology has exploded, and now there's light everywhere. And so there's all sorts of super interesting studies going on that are absolutely mindful of clinical advances and how it might help patients in the future. So I'm actually very optimistic.
Yeah, I'll piggyback on that. I agree. I think there's been there's been many advances in the last couple of years. The development of technology and our ability to learn about metastatic breast cancer in the laboratory has just exploded recently. And I would say in the next several years, we're going to start to be able to translate those findings and that technology into the clinical setting. And I'm very hopeful and very optimistic that there are going to be a lot of new discoveries, new treatments, new ways of classifying breast cancer that will help enable the promise of precision medicine. So I share that optimism. It's still hard, and it's still going to take time. And we wish there were answers for every patient with metastatic breast cancer tomorrow, and it's frustrating that it does take time. But I think we continue to move in the right direction. I'll say one last thing about the MBC project. Thanks, Jean, for that plug, which is this is a way for patients around the US and Canada to be able to participate in this type of research. It's not clinical research, but it's the opportunity for patients to be able to share their information, share their samples, their tumor samples, their saliva samples, their medical records, their stories with the research community generally. And we started this about 5 years ago. We have thousands of people, over 5,000 people who've signed up around the US and Canada with metastatic breast cancer. And in the time of COVID-19, where research and traveling is hard, I'm hopeful that this is a way for people to be able to continue and the research initiatives to be able to continue, because remote research is something I think that we're going to need to learn to do more of. So I would encourage you to go visit mbcproject.org, and you can learn more about it. And our team would be happy to field any questions if people have any.
Thank you so much, Dr. Wagle and Dr. Carey. I am always so grateful for the amazingly smart, compassionate individuals that choose to go into this field. And we know at Living Beyond Breast Cancer, this pandemic has really hit our healthcare professionals harder than anyone else. So just holding you all in the light and thanking you for continuing to do the work and continuing to really care and keeping us all safe.